today's training webinar, I'm going to present you or give you an introduction into multi-omics pathway analysis, focusing on our tool that is specific to this problem called ReactomeGSA. So as mentioned by Anna before, this training is recorded and put on YouTube later on. And as you can see, this is just a YouTube search from today. We have a similar training from last year. The first half of this is essentially completely new. So I'm going to show you new content, new ways to do stuff. But the examples we're showing, they are the same thing. So if you watch this on YouTube or if you've seen the first part, so the examples are still the same, but the first part is different. Also, since we've recorded this before and I've shown the same examples before, I will be going through them rather quickly this time. So if you feel that something is missing, maybe watch the video from last year first. But of course, you can also contact us. And this is the most important slide now. If you have any questions, any issues using the tool or questions about pathway analysis in general, please send us an email to the Reactome help desk, which you can reach on the help@reactome.org. Okay, so this is today's schedule. We'll start with like 2 introduction parts. The first one is a very general introduction into pathway analysis overall. And then I'll be talking about Reactome and ReactomeGSA just to give you the context, and then basically it's just tutorials. So I'll show you how to analyze your own data. This has been a huge question over the last year. We've had a lot of support questions using 2 methods, the Geneset analysis and the Geneset variation analysis. And then come the examples. So these 3 examples, they are the same that we had in the last year because they give you a very broad overview of what you can do with our tools. But I will be going over them rather quickly, rather briefly. So if you have questions, either watch the video from last year or just send us an email. So short introduction to pathway analysis. So the most common algorithm and approach that you probably see are so-called overrepresentation analysis. And this is what most web services provide you with. Examples are Reactome, Panther, or David. You can recognize them that they have essentially always kind of an input box where you just copy and paste your identifiers, and then you get your pathway results. So the advantage is really easy to use, super fast, but it has a big disadvantage. And that is it ignores your quantitative information. If you use these tools, you have to do your analysis, statistical tests, overrepresentation analysis yourself first. Then you select your significant genes and you copy it into the tool. So the tool doesn't know about any quantitative regulations in your data. This reduces your statistical power. It also means that you have to set the significance thresholds on the gene or protein level. And you need separate analysis for up and down regulated genes and proteins. So you can't just copy everything at the same time because the tool doesn't know what happened. In the background, so the math behind the thing is essentially it's a hypergeometric test. The problem it solves is this like 4-square table. So you have the genes that are part of a pathway, the genes that are not part of a pathway, and then the genes, the input genes, right? The ones that you just pasted in this box, and the background genes. And this is something you will never see most of the times because these background genes means normally the tools use all the genes that are annotated in their pathway database. But of course, this is not completely correct, since if you work in a specific cell line, maybe not all the genes are expressed in that cell line, and the background would have to be adapted, but the tools actually don't give you this option. So the results are, let's say, more or less accurate. Now, Geneset analysis and all the stuff that I'll be talking about today and showing you is this class of algorithms. They actually use the quantitative values as input. Sometimes they're rank transformed, but they always use the quantitative information. It normally performs a 2-group analysis directly. It doesn't need a background, which is great because it doesn't matter where your data comes from. The statistical assumptions are more or less correct. And it retains the quantitative information. And if you plot this, this is the problem that's solved here, right? So you have all genes of a pathway. It's basically what the plot shows. So you have your genes, all the genes of a pathways, but you have your 2 groups of samples, your 2 phenotypes that you compare. And then you get this kind of distributions of the expression values, and you can do a t-test, for example. So most of the tools actually use some kind of t-statistic, and you get a p-value per pathway. Now, the last set is probably the least known class of pathway analysis approaches, algorithms. These are so-called Geneset variation analysis. And what they do is they map expression values on the pathway level. So you don't need to define any groups beforehand, because the analysis algorithm actually doesn't do any differential expression analysis or differential analysis here. It just maps the expression values. And this enables pathway level downstream analysis, for example, like comparison with continuous parameters. I'll give you an example. The class before can do 2 group examples, but what if you want to check whether a certain pathway correlates with your participant's weight, for example, or your height, or any other, or survival time, for example. All of this cannot be done with a simple 2-group comparison. So here we want to use this. Exactly, so I just drew here a simple diagram. So what happens is you have an input, you have all your genes or proteins and the samples, right, the expression values. And what the Geneset variation does now is it maps it on the pathway level. So as a result, you get per pathway and sample 1 expression value. But we're going through this in the tutorial in a second. So what's Reactome? What's ReactomeGSA? So Reactome is a pathway database. Reactome is quite special because the pathways that you see in these diagrams, they are actually created by PhD level scientific curators. So these guys, they have a topic, they try to read all the papers on that topic, and then they draw the pathway diagram. Now the overview in Reactome is here in the top left. This is kind of this hierarchical structure. This hierarchical structure is manually curated. It's a bit artificial, right? That's what the curators thought is the best way to curate this information. The next level, sometimes there are these great diagrams. These are actually drawn by a professional illustrator who creates these nearly publication-ready plots. These are all interactive for the pathway levels. And here you can already see this is manually curated, right? And on the lowest level, if you really zoom into this, you then get these reactions. And that's where Reactome comes from. We see, compound A is phosphorylated, is interacting with compound B, and then, this new compound exists, and there's an ATP transfer, and so whatnot. And this is actually what the curators curate. So they build these pathway diagrams by hand. Now, what's ReactomeGSA? Now, ReactomeGSA was created because we felt that something was missing, right? And that's kind of this multi-dataset problem. When you today analyze data, let's say you do your study, right? You test some drug in a mouse experiment and you do proteomics. But that's not, of course, the end of your story. Now you want to compare your data with other published data. And as soon as you look for other published data, at least that's what happened to me, you see, okay, wow, there's this cool other mouse study, they do the same thing, but they use transcriptomics. Maybe you want to compare to human data, but here this is an older, old microarray data set. And now immediately you're in this struggle, right, that you have to combine different omics technologies, maybe even different species, and you want to get one result. And that's what we try to solve with ReactomeGSA and made as simple as possible. So what are the key features of ReactomeGSA? So first of all, it's a multi-omics tool, meaning it automatically chooses the right statistical model for your input data. So as soon as you tell us what the data is, we choose the statistical model. It highlights the similarities and differences between these omics datasets. It's a quantitative Geneset analysis approach. Actually, it supports multiple ones, which means this increases your statistical power, and it shows you a direction of the pathways, right? So we can tell you if your pathway is up or down regulated. And again, it supports different omics analysis approaches. So interactive visualization, as in our opinion, a very important part. So Reactome has this great web interface. So this allows you kind of an intuitive data analysis. It's kind of a visual comparison of multiple data sets, and you can really, preserve the quantitative information and zoom into the details. All of that, I'm going to show you in a second. It's platform agnostic. Now, platform agnostic here doesn't mean based on your omics technology, but actually on your analysis system. So we try to combine all the data that you put in the web interface or our R package, you actually can swap between them. So I'm going to show you that as well. So you can jump from your R session into the web interface and from the web view to the R session. And we wanted to really interface this with public data. So to simplify the reuse of public data. And automatically import datasets from key resources. So that's kind of a diagram of how the tool or this whole ecosystem looks like. You can load public data from Expression Atlas. You can upload your own data in the web interface. In the background, the web interface is actually using a separate analysis system, which is the heart of ReactomeGSA. And then it provides you this interactive pathway view and also gives you access to PDF and Excel results as reports that can be sent to you via email if your analysis takes a bit longer. So before I start, I'm just having a quick look at the questions. So there's a question, will you record the presentation? Yes, everything I say is recorded and will be put on YouTube, but that's already answered. Okay, sorry. Okay, sorry, I'm speaking too quick. I'll try to slow down. About the p-value calculation, I'll come to that later. Okay, so a very good question here right from the start is, how is ReactomeGSA concerning microbial pathways? Now, this is very important. Reactome is curated only essentially for human data. So human data is the basis. Several species are automatically inferred through algorithms, meaning the identifiers, the human uniprot identifiers for proteins, are mapped to the respective species in an automatic process. This means that, yes, we do support mouse identifiers or Drosophila, but the pathways, so the curated data, the source data, is still the human data. Okay? So meaning that in ReactomeGSA, we're actually going so far that we only report human pathways. It doesn't matter whether you put in mouse or anything else, you actually get a result, the human pathways, because that's what actually was curated. Okay. There's a question, how do you correct p-values for multiple testing? Quite simple. This is the FDR Benjamin Hochberg approach. Are the pathways in Reactome experimentally proved pathways, or only does this include predicted interactions? So Reactome pathways are based on literature which is experimentally proven. So nothing here is automatically inferred or predicted interaction. This is all curated, published data. And in Reactome, you can actually click on the publication that was the source. So now I'm going on and I'm starting with the first example. And that's the question is how to analyze your own data. I'm going to show you two methods here. We'll discuss how to choose the right analysis method, upload your own data, use the interactive pathway visualization, and store the data. As you can imagine, this is like all days about ReactomeGSA, so this will take now the longest, and please answer questions, and I'll make breaks in between this section now just to answer them like I just did now. And I'm now going to actually close my screen here, sorry, to open the Jupyter Lab. So I'm just going to do this now, and we'll start this now live. So as you can see, I've prepared here a data set, and this is the output. I started with proteomics because we saw in our kind of last year in the mailing list that most people had issues with proteomics data. So what you can see here, this is kind of the output of a proteomics pipeline. It's called FracPipe. It doesn't matter whether you have proteomics, microarray, or transcriptomics data. I just chose this because it's the most complicated example, but it's the same thing for everything else. So what we need as input is what you see here on my screen. It's a real data set. So in the first column, this is just open now in OpenOffice. You have the column with your identifiers, and then for any subsequent column is the expression values of one sample. And what you see here is this kind of anonymized. I had to anonymize this because this is patient data. So we have sample identifiers here. Now I'm just going to shorten this. So 1, 2, 3, 4, 5, 6. We'll just keep the 6 samples here, and we'll just delete the rest. As you can see, it's quite a large data set, but this is just for demonstration purposes here. So I'm just going to delete that. And you can do all of this in Excel, and I actually recommend you have a look at your data before you send it to us, because that can save you a lot of time in the question. So now we just have 6 samples here. And I'll start with this, because this is really the stuff that where we got like 3 questions a day, or a week, let's say this. So the issue is, if you look at this column, you see these strange identifiers. Now, for those guys who work in proteomics, you will recognize this as a combined Swiss prot identifier that was extracted from a FASTA file. What it means is that the actual accession number, right, this could be equivalent to a gene name, is actually in the middle here. Okay, so this is not a valid identifier. And if you upload this to ReactomeGSA or any other tool, you will get an error message. So please do check that this is actually the correct identifier. So here is not. I just saw this, so I'm now adding 2 columns here. I'm just going to quickly show you how I solve this issue. So I like regex, so I'm going to do a regex here. Sorry, regex. We say the input is the identifier, and now I'm just saying, okay, only extract between the 2 pipes or dashes. So I'm just saying, okay, ignore that part, extract everything that is not a dash. So this is recorded, so you can look at this. If you're unfamiliar with regex, you don't need this for the tool. But just to show you that this is really an issue, sorry, I just mistyped here. So, and now we replace this with everything that I extracted here. So, okay, that was the wrong one. Let's see, we have to add 1 dash here. So now I double check this, right? This portion that was just extracted here is actually the identifier between the 2 pipes. I can just double click here and it's filled for all the identifiers. I can just copy it because this is a formula. I copy and paste it here. Sorry, that didn't work. Control Z, I need to use this paste special command as unformatted text, and we'll just fill it here. And now what I can do is I can delete these 2 columns, and that's really important because we need this kind of file where we only have the identifiers and the samples. In a lot of transcriptomics datasets, you get columns where you have an identifier in the first column, second column, the gene name, third column, the gene symbol, for example. In these cases, really format the data like this. 1 column for identifier, and here please use the most specific one. For transcriptomics, it would be, for example, ensemble gene identifiers, not the gene symbol, but we map everything, so it doesn't matter, but just to give you more precise results. And then you have the samples. So first is the sample identifier, and then all the sample expression values. So I'm just going to save this now, save as, and if you save it, you need to save it as a text file, right? So here I'm in OpenOffice, but in Microsoft Excel, it's the same story. Just text here to CSV file. It doesn't really matter whether it's a comma separated file or a tab delimited file. That's automatically recognized by our tool. So I'm just going to close this now. So this is our file here. And this is now the Reactome website. If you enter it, the URL is reactome.org. You click on analysis tools, and this opens actually a complete web-based, browser-based tool that's called the Pathway Browser, where we have different tools. So the analyzed gene list is your classical overrepresentation analysis, where you just copy and paste the identifiers. And ReactomeGSA is hidden behind this button called Analyze Gene Expression. Now, in ReactomeGSA, we have currently three supported algorithms. PADOC is the one we actually recommend. This is a classical gene set analysis approach that has the nice feature that it downweighs shared genes. So some genes, some proteins are part of many, many pathways, right? And in this case, we don't want them to kind of dominate your data set. So PADOC is downweighing them in the statistics. We also support camera. Camera uses linear modeling, which is comparable also to a t-test, but has the great advantage that you can add covariates to your data. That could be, for example, if you have a clinical study and you measure something in patients, but the patients come from 2 clinical centers. You could say, okay, I might have a confounding parameter, the clinical center, and I want to regress this out of my data. This can be done with camera. So in camera, you can then say, okay, the clinical center is my covariate, please regress it out. And you can add any number of covariates. PADOC supports this to a certain extent, but not as well as camera. SSGSA is a gene set variation analysis, and I'm going to show this to you later. So we'll select PADOC. Here you have this kind of, dial symbol where you can change the parameters. So the parameters here is use interactors. And this is something, one of the questions that came up about the prediction. So if you enable this option, what's happening is we use protein-protein interaction data from intact to enrich the Reactome pathways. Since Reactome pathways are manually curated, we don't have information for every gene or protein, but here you can automatically add close reactors or interactors to the pathways automatically. So I'm not going to do this now. I'm just keeping it simple. Everything else I'll leave now as a default, and I'll just continue. So here you need to click the Add Dataset button, and and here you select a file from local folder, and you have to select your dataset. That's how we know which omics technology you used. The options currently are RNAseq, where you can submit raw counts, which is the recommended option for RNAseq, but also normalized counts. Sometimes some core facilities, some published papers, they only provide you, for example, TPM transformed counts. Then you have to use the normalized ones. Proteomics, we support the intensity-based normalizations, which are the most common ones, but also spectral counts should they occur. And microarray data, important microarray data has to be normalized before you submit it to us. So I'll just take here my proteomics intensity data. Let's just click this file. I'm just taking this from my recent files, CSV, and we're done. So the file is uploaded, and as soon as the upload is done, you could come to the annotation window. So here we can give a dataset a name. So for example, drug test. And now you have to add some metadata. Now the metadata, we just click, maybe we call this treatment. And now we call this, they got the drug, control. So I'm moving to the next one with the tab on my screen. So I just moved to, so I'm done here. I click continue. And now you have to say, okay, what's your comparison factor? Comparison factor is essentially the parameter, or the variable you just entered that you actually want to use for the comparison. So here we say treatment, we just entered. First group is the control, second group is the drug. So here, the first group is always the baseline, okay? So anything that changes, for example, upregulation means based on the first group, it's upregulated in the second group. So we click continue. And that's it. So now we added a data set. So as you can already see, you can add multiple data sets, but that's for later. So click continue. So if you put your email address here, you will get an email with the links to all the results as soon as the analysis is done. This is helpful, for example, if it's a very big data set, you can also close your browser while the analysis is running, and it will not stop. So I'll just click go now, and the analysis is running. So this is actually happening live. I didn't prepare this, so I didn't use prepared data, just to show you essentially how it works. The data is submitted. You see the progress being displayed here, and then the result is in. So unfortunately, Zoom is having some issues. So I'm just jumping back here to the presentation. I'm clicking on present. So did that work? Yes. And now I can open the Q&A again, just to keep in touch with that. So, okay, I'll quickly look at the questions. Sorry, I have to move close to the screen so I can see this. Can we do this analysis with any annotations such as gene symbol? Yes, we support, I think, 20 different annotation types. Just put it in, we most likely support it. If not, send us an email, probably we'll be able to fix it. Next one, what do you use for the analysis RNAseq data, TPM, RPM, or ReactomeGSA on ReactomeGSA? So this is kind of a mixed, Christina, this is kind of a mixed question. So TPM, RPKM, these are normalization values, right? So we recommend you to upload your raw counts. So the actual raw gene level transcript or read counts per gene, because this allows us to have more accurate statistics. If you only have TPM or RPKM, yeah, we support them as well through the RNAseq normalized, but recommended is the raw counts. So this is done. Can this be applied to CRISPR-Cas9 experiment? Well, it depends on what you measured. So CRISPR-Cas9 experiment, if you are more specific about what you measured as a readout, I'm happy to answer it, but this has to be more specific. Gene set enrichment is performed at the gene level. How do you solve the multiple isoform values for a single gene? Which one do you choose? So Good question, and I'm going to use this actually to answer a different thing as well. So what we do is we use your identifiers, okay? So whatever you had, whatever level you annotated it. So what we need from you is already gene or protein level data. For proteomics, this means actually doing the whole protein inference. That's something you have to do. For transcriptomics, it's easier because in your alignment, you already chose whether to align to only the genes or add isoforms as well. And for microarray data, when you have multiple probes for the same gene, you have to take care that you only report values for a single gene, because as the ones of you who work with microarray know, there are multiple ways to do this. That said, what we do is we then check to which pathways a gene maps, because what we do is, of course, we then map your identifiers. Now, if you, for example, use gene symbols can be quite ambiguous. So we have the same gene symbol for actually a few genes sometimes, or isoforms as you put here. In that case, what we do is we check to how many pathways does it map, or how often does it map, and then we have this kind of 1 to n phenomenon. So one entry might map to 3 entries in our backend. And what we do then, we recognize this and the tool corrects for it. And the expression values, since we're only interested in values for our statistics, are only added once. So this is taken care of. But all the isoform stuff has to be done in the pre-processing pipeline. How about metabolic data? Is it possible to use Reactome for the analysis? No. Unfortunately, for metabolomics data, we are not there yet. Curation is ongoing, but we haven't reached a critical threshold yet. So what can we use? DNA methylation data. We're working on that. Okay, so DNA methylation data, send us an email if you have some data. We'll probably find a way to do it, but not in the web backend at the moment. Metabolomic is done. Integrate metabolomics, transcriptomics, unfortunately not. Okay. So I'm just keeping this quick. The background is not used in these approaches. Like I showed you, we're only looking at the different distributions. So I'm just going back now to my analysis that is running that you're seeing. So the analysis is done in the meantime. This has been done probably for a few minutes already. So you see two things here. First of all, you see a list of the available reports. We will keep them for later. And here, this first button click to visualize the results in the Geneset analysis. That's what we're going to do. So what happens now is, here in this lower part, you only see our dataset name, which we call drug test, and you see this Reactome pathway overview. And the color scheme is very simplistic. So we have significantly upregulated, not regulated, downregulated, significantly downregulated. And as you can see, there are some yellow pathways. Now, let's say I'm super interested in the role of phospholipid in phagocytosis. So what you can do is, so I'll move my mouse here to see it. I'll double click here. And what's happening now in this web interface is it's actually zooming into the pathway with this very nice animation. And here is a curated pathway. But that's actually the raw data. And the very nice thing is that now you are at the gene or protein level. And you see for each of these entries, so that these kind of boxes, these are compounds. They are made up of multiple proteins. And these circle-ish things are single proteins. And you can see how many proteins were actually found that are part of this compound from your data set. And you also see whether they're up and down regulated. And if you zoom even further in, so I'm just going to do this now, so you can kind of drag this here, which is a bit slow. So I'm just zooming in, this is just with my mouse wheel, you see, you get more granularity. And you can see that some of the proteins were actually downregulated and some up. Now that's just a biological truth, right? It's not a 100% perfect result, but you see there's some difference. And if you click this kind of triangle here, you actually get the list of proteins that were found in your data set and whether they're up or down regulated. So this tool here gives you, I think it's quite unique, the great opportunity to look at the large pathway level and then drill down to the single gene and protein level and actually see your expression values mapped onto these pathways. And since everything here is under the Creative Commons license, you can use all of these diagrams for your publications. Another cool feature is here, up here, is called the Voronoi visualization. I always struggle with. It's called Reactome. And this is kind of a different visualization, as you'll see in a second, to use more space on the screen and give you a better view. And what you can see here, all the pathways again follows the same hierarchical structure, but now they have these kind of squares, and you see immediately which of your pathways are up or down regulated. It's the same story as before. So I can cellular hexose transport, let's say I'm interested in that. You can double click it, it zooms in and then gives you the pathway diagram. I'm just going to close this now. So a nice way, so I'm more of kind of the reading kind of guy, so I prefer to look at tables. You have the tables of your pathway here. You see here upregulated, 2 errors up means significantly upregulated, 2 errors down significantly downregulated, and 1 or the other just means non-significantly up or downregulated based on a FDR threshold of 0.05. So you see immediately when I hover with my mouse over one of these entries, the upper view also highlights this pathway in the diagram, and you can then drill into the pathway. So you can see here, so here I have the NMDA, I double clicked here, and now it's zooming in. It also tells you how many of the entries were found in your dataset. Now the reports, you have 2 download buttons, and don't get them confused. So the one I'm talking about is here on the left. See, here on the left, you have these 3 buttons, the results, identifiers that were not found, so stuff that I submitted that is not annotated in Reactome, and then the download section. I'm talking about the download sections. And here you have the reports that you saw before. So the R script, the PDF report, and the Microsoft Excel report. I'll start with the PDF report, so I'm just clicking here. And this gives you kind of a nicely rendered, at least I believe so, report of your analysis results. So you have this kind of volcano plot of pathways, and then a list of the top regulated pathways, the name and the identifier, and whether it's up or down regulated with an FDR, average fold change, and the number of genes. And if you have multiple data sets that you submitted in your request, you will have multiple columns here and can immediately compare the results between your data sets. The other one is the Microsoft Excel report. Again, I'm just clicking here. This opens our Microsoft Excel file that gives you the pathway result if you are familiar with R, for example, that you would expect kind of if you do the pathway analysis in R directly, the name of each pathway, the direction in this data set, the FDR, the original p-value, the number of genes, and all the different parameters for the statistics that was used. I'm closing that again. Now, why am I kind of stressing on these results? Reactome has a 3-month release cycle. So about every 3 months, we release a new version of Reactome. And since it's impossible to keep this website running with multiple versions at the same time, your results cannot be stored that long. So the results you see now, they are generally stored on our servers for, depending on usage, a few weeks, but then they are removed, right? So you cannot get the same result again. And every 3 months, a new release is out, and you actually will get slightly different pathway results, because a new release means the curators are working non-stop, new pathways are curated, some probably with more details, and of course this changes your stats. So if you need reproducible results because you're working on a paper and you want to update your figures, keep that in mind, that you somehow store your result using one of these options. Now, the most elegant work option, at least for ReactomeGSA, is to use the R script, and that's what we're quite proud of. So I click this button now, and what's happening now is this is just opening a text file. And this text file is an R script. So what I'm going to do, just to prove this to you, is I'm just going to copy this. And I have here, I'll just open an R session. So this is now a plain R session on my computer. It's running now, and it's okay, it's R 4.1. I'm just copying. So this is exactly the same text you saw before. And I'm just going through this line. So what it says is, if you don't have the ReactomeGSA package, install it from Bioconductor automatically, load the package, download the result, and this is your unique identifier of your results. So this is unique to you, and immediately save this as an RDS file and then get the pathway. So I'm just going to execute this now. So as you can see, okay, I've got the ReactomeGSA installed already. So it's loading the package and it already got all the results. So now I have the all pathways objects in my namespace. I'm just going to show you this. Let's do a head command here. So I'm in my R session, and what you see is exactly the same table that you saw seconds ago in the R session, in the Microsoft Excel file. I can even do the plot volcano command here with my result object. Say enter, and you get the same volcano plot that you saw in the PDF file. This is one-to-one the same data. And in the R script, we actually put in this save RDS file command. So immediately the file is saved here. And this is kind of your source data, right? So you can load it again. And when you need to update some plots for your figures, you can use this RDS file on your computer to upload your plots if you do them in R, for example. So this is quite nice to kind of immediately jump into R and continue working there and have your results there. So I'm going to use the time now. So the questions are coming in. Please vote your questions up if you're super interested in them. Let's see that. So I just have to quick. So can this be applied to CRISPR-Cas9? Okay, please. I need more specific ones. Proteomics data must be raw or normalized. Okay, the proteomics data. So raw proteomics data is an issue anyway, right? In proteomics, you generally measure on the peptide level, at least in MS-based proteomics. And when you do the protein inference, so you get from your peptide level data to the protein level, you should have done the normalization already. So proteomics should be normalized on the PSM level. For those in proteomics, they will know what it means. So you generally should send us normalized data. Is Reactome available as an R package? So yeah, I think this question is answered now. Can we use ATAC-seq chipset called peak adjacent gene dataset? So we managed to do some of these analyses already, yes, but it's not implemented in the web interface or the R package automatically. Do you think we can treat as proteomics the phosphoproteomics data? Okay, so that's really important about phosphoproteomics. So yes, you could, but of course, Reactome is phosphosite agnostic. And if you do phospho, it really makes a huge difference which phosphosite at which location is differentially regulated. That's kind of your key information. And that kind of positional information would be lost here. So yes, you can use it, but you will lose a lot. Next one, to study network topology, each partial, can I use Reactome FI network as a base extract from the genes? So I'm not discussing Reactome FI here. Please send us an email. It's just a different tool that we have. How's the curation status for glycan information? Not great at the moment, so it's not complete, far from complete, unfortunately. Is it more correct than HR or DSEQ2 to calculate fold changes or DGE if we use raw counts? So in the backend, we actually use limma. And like if you use HR or DSEQ2, you know that HR and DSEQ2 want raw counts as input as well in transcriptomics. So we do the same thing on our end. Don't forget, we do all the analysis on the pathway level, not the gene level. So yes, always try to use raw counts as input. In the HR and the DSEQ2 vignette, they actually stress this quite a bit. In case of partial gene shed in the original data, what would be the ideal metric to consider the pathway quantification provided by Reactome? So I'm not quite sure. Can you say, Sunil, if you ask about the incomplete pathway gene sets, I'm not quite sure what you mean. So if you post the question again, I'm happy to answer it. And the last question I'm going to answer in this break, do you think we can treat this proteomics, the phosphor, sorry, I have this already. Lipidomics information is unfortunately currently not directly supported. Okay, so we are back here. And now I'm jumping back to my presentation. And we're jumping, so what we discussed just is you choose the right analysis method, paddock, if you have this kind of very neat two-group comparison, camera, if you have some covariates you want to regress out, how to upload your data and how to really, I'm stressing that, prepare it, and the interactive pathway visualization. So I'm coming to the next thing, and that's kind of this SSGSA approach, where we had a lot of questions where people were kind of struggling to know why they would use it, and it's really powerful. So I think this is underused at the moment. So what I'm going to do now is I'm just jumping to my Reactome window view again, and I can open the analysis again by clicking the analysis button here on the top. So this comes back to the screen that you just saw before. So I have a back button here. So I click back, click back, click back. And now what I'm doing is I'm just clicking SSGSA here. Done. I click continue again. The dataset is still stored. So I'm just keeping that as it is. I'll keep that as it is as well. So we want to create reports and click go. So as you saw, this is quite neat. You can just play around with this, check how does camera perform, how does PATOC perform if you have a two-group comparison. And here actually the group comparison parameter that you entered, so here it's drug versus control, is completely ignored, since SSGSA now maps my expression values on the pathway level. This is the easy part. The more interesting part, and that is how to analyze this data. So it's already running, so we got the results. As you see, the reports are generated, but we can already look at the results by just clicking this X button here. And now, what you can see is this lower control here. This looks like kind of the control in a video player or something, has one of 6 options, and you can see each of these options is now your sample. So I had 6 samples, and if you click here, I move between the samples. And if you look at the screen, nothing changes. It's not a bug, just the fact that the visualization really doesn't work well for this kind of data. We still offer it since it's like part of our software system. But the thing you are interested here is something else. The thing you're interested in here is actually the reports. So I'm going to open the analysis window here again. Okay, the reports are not created yet, and this is now a bug. So what I'm going to do is I'm just going to launch this again and redo the analysis. So I'm going to Geneset analysis. I'm doing SSGSA again. I'm adding my data set. So I have proteomics density data. It's a CSV file. So this is uploaded. So we're just repeating the steps that we had before. I need to add an annotation. So I'll just keep this as A, and then let's say B. This is has reasons for the interface that you have to select this, even though it's ignored, because it just made our life a lot easier. And we just click OK. So the analysis is running now. I'm using this time now to answer a few more questions. So is there an R package available? Yes, it's called ReactomeGSA, and I just showed it a second ago. So I would like to know, is this platform working with microRNA pathways? So microRNA pathways per se. So microRNA is some microRNA might be annotated. Now I'm lying if I say yes here. So if microRNA is part of a pathway, it's annotated. Let's put it that way. How big the coverage for microRNA is, I don't know at the moment. How you identify the pathway is up or down regulated, but you have multiple genes as well, both up and down regulated inside the pathway. So this is a very good question. So the question is, what do you do if, as you saw in my example before, right? Not all the proteins are up or down regulated, even though they're part of the same pathway. And that's just a biological truth. So the simplistic answer is that we just take the mean, more or less, but we actually take the mean fold change to report the pathway level fold change. In camera, it's a bit more complex for the algorithm, because here we can actually use the t-statistics that we get to kind of also incorporate the regressing out of these parameters. So that's how it's done. And the simple answer is it's essentially more or less a mean with some statistical quirks. How does it calculate up or down regulated? That's what we did. Can it deal with biological replicates? Absolutely. So biological replicates, that's supported by PATOC and camera. Yes, you can add them as kind of a covariate, and in camera, this is perfectly implemented. And I'm also going to show you how this works. So the report generation just takes a lot longer than I expected. So here I did cheat and I actually prepared the data. So I'm just going to jump to the session now. So we stick within the time. And this is, I think here, the GSVA example. So essentially what I did here, let's stick with our example with the 4 samples, okay? Just imagine that we have a weight for our patients. And we have these random weight values. Now, this is a copy and paste of my previous analysis, and I can actually run this again. So your analysis identifier stays valid for a few weeks. I can't give you a exact time frame because this is automatically done per usage. So now it's downloading the result. And the result now looks like this. So this is all pathways. So what you see here is I have my pathways again, but now as my columns, I have all my samples that I just uploaded. And for each sample, I have one expression value. Now, one thing I can do now is I can now correlate my pathway expression values with my weight, so my fact of interest. So this is some R code. So what I do is here, I run this for every row here. I get my pathway expression values as kind of as numeric function. I run a correlation test here, very simple test in R. And I just say, okay, return the p-value and the estimate. So I'm going to run this now. Oh, great. I forgot to add the weight. And let's add this as well. So, and now I have the correlations. So I'm just converting this to a table. I'm filtering this, say, give me everything that is p-value below 0.05 and just show the first 4 entries, and that's what you get. I get my pathways. I know how well they correlate now with weight, okay? Now, of course, important, you have to correct for multiple testing. I didn't do that here, so I'll fix that right away. We can't leave it like that. So let's say to correlations, p-adjust is p-adjust. This is really great in our correlations, p-done. Now we say, of course, our adjusted p-value. So we'll just copy this and put it here. Has to be smaller. Let's see. Oh, and we have nothing significant anymore. Bad example. Let's see where we have something. We don't know. So let's just take some random stuff. Okay. So, of course, this kind of failed miserably. Sorry about that. Just keep it at the p-value here for the sake of this tutorial. So use multiple testing. As you see, it can change your results dramatically. So we'll just keep these non-significant results, as you see. Adjusted p-value is 1, so this is nothing, but we'll just keep it. And now we can, for example, take the pathway expression. So we say, okay, this pathway here, just take it from the all pathways matrix. You take this pathway, 2 to 7, these are my columns of my samples. And then we can plot this against weight. And you have this pathway expression value and plot it via weight versus weight and see if there is something in your dataset here. You can also have this nice heat map. This is called plot GSVA heat map, where you have all the different pathways over all the different samples and see how their expression changes between your pathways. So this kind of tool is really, really great if you don't have specific phenotypes for your samples. So if you don't have your 2-group comparison, if you have continuous parameters, for example, survival time, weight, everything that's continuous you can do with this analysis, but the statistics has to be done by you. So we just provide the pathway level expression values, but you have to do the stats and actually get the results. As you can see, I'm a fan of R, so I do everything in R, but this can be done in MATLAB, even in Excel, it doesn't matter. This is not the limit, and you can download this as an Excel file as well. So you see, in the meantime, we actually got the results from my example as well. So I'm just going to show you how the Excel file looks like for this result here. So it's the same thing. You get the name of the pathway, and then for each and every sample, you get the intensity values or expression values for that pathway. Okay. So I'm now going back, and we are now at the ReactomeGSA R package. So I'm just checking time now. Okay. So the R package kind of is the thing that you just saw me clicking around with. The R package also allows you to start your analysis. Okay. So you can just load in summarized experiments, DGE list objects, E-list objects for microarray data, We have this add dataset function that does the same thing like the add dataset here. You can add multiple datasets, and then you get your correlations. I'm skipping this example due to time limits, but you can find a recording of exactly the same sample showing you how to use data from Expression Atlas in the YouTube video that I showed you before. I'm just showing you how it looks like here with a TCGA analysis that I did. I had to do this beforehand because it just takes too long. So if you know TCGA is this huge effort to characterize cancer samples, you can download the data easily using R again. So here I downloaded this data for example, for here the lung adenocarcinoma data. I get this kind of as a DGE list object. By the way, we have these Jupyter notebooks on GitHub. You can find them on the URL at the end. And you get this DGE object. And now it's really great because you can just load our library, the ReactomeGSA R package. You create an analysis request object where you supply the method, paddock, like we did before. You can set additional parameters, and that's kind of the options button I showed you in the web interface where I said, okay, use data actors, include disease pathways, yes or no. And then you add the data sets, and you can add multiple data sets. So here, for example, I can now use as expression values directly this DGE object I had before. And the cool thing is that you have all the metadata readily available. So you can say, okay, this is TCGA lung, it's RNAseq counts, and I want to compare this TIPB group, which I created randomly. This is B cell high or low, and you compare high versus low. And then you run your analysis with this perform analysis command. And as a result, and that's kind of the most powerful part of ReactomeGSA, you get your pathways, the regulations, not for one data set, but for all at the same time. And for example, this is kind of a data set where I check for high and low numbers of B cells, something we're studying. And you can see that samples with a lot of B cells here, melanoma, they have a lot of B cell receptor signaling. That's what you expect. The same is true for ovarian cancer, same is true for breast cancer, same is true for squamous cell carcinoma in lung, but it's not true for lung or adenocarcinoma. So apparently the role of B cells in lung or adenocarcinoma is completely different than in the other cancers. And the analysis here, well, of course, runtime was long, but it took like 20 minutes, and you have this multi-dataset analysis. You can add multi-omics data. So TCGA has this sister project called CPTEC, where they measure protein level abundances. Here is the code to download the breast cancer data on the transcriptomics and the proteomics data. I again have the same groups, lots and less B cells. And what I get now, I can have these nice plots here, the fold change of pathways in my data sets. And you can see most of the pathways have, well, quite similar fold changes, but there's a group that's downregulated in the CPTEC, so in the protein level data, but upregulated on the transcriptome. So this is quite puzzling, of course. I can immediately investigate this. See, upregulated in breast TCGA. So this is transcriptomics, but downregulated on the protein level, all significant. And these are really important things, like, for example, IL-20 signaling, IL-7 signaling. So all of this is really important to anti-cancer immunity, and they have different regulations on the protein and transcriptome level. Finally, the ReactomeGSA R package is the most used function, actually, has this neat function to also analyze single cell data. We support single cell experiment and SARAD objects directly. Just say analyze SC clusters. It's one command, it does everything. And what it does is it creates kind of from the cell clusters. So here again, I'm B cell focused because that's my research interest. Different types of B cells. I don't know what they are. So I'm just can now run this, analyze SC clusters. And now, for example, B cell receptor signaling again, I see that for my different cell types, there's a big difference in this B cell receptor pathway. So it's a neat way to get pathway level data into single cell data with, I would argue, the least amount of effort. So again, you see the heat map here, downstream signaling of BCR, which I just showed you before in a color coded, but also different NF-kappa B activation. This shows a big difference, for example, in this double negative group compared, for example, to plasmablasts. So this gives you an option to get the pathways into single cell data. So in today's webinar, I hope I could show you how you can use ReactomeGSA to do your multi-omics, multi-dataset pathway analysis using the pathway browser, but also the ReactomeGSA R package. We support multi-omics experiments, we support multi-species experiment, and everything is done on the quantitative level. As a result, we offer you this kind of quantitative pathway level view combined with our interactive web interface. So I'm just looking at the few questions now. Now I know I'm not able to answer all of them or even half of them. Please send us the emails. So if I know a lot couldn't be answered, please send an email to help@reactome.org. I'm happy to provide support and we are happy to help you. So I'll just give you a quick look at the answers. Can you access gene lists for individual groups to back them down upregulated genes? Yes, this is what I showed you. I'm just going to do that. Sorry, acknowledgements is important, very important. So if you double click here, sorry, I have the wrong example here. So normally, if you double click on a pathway and you go into this kind of very detailed pathway view, you get this box. So this is loading up now. Let's scroll in. You have this small triangle here. And if you click the small triangle, you actually have your identifiers that were found. Okay, and together the expression values. That's Reactome work for RNAseq from gut microbiome data. So depends on what kind of data you have. Microbiome data would be the data of microbes or sRNA. So I'm not quite sure what you're asking here, but if you are looking at the transcript changes of the human gut, yes, we can do that. Can you perform all the analysis in the R package or just the results gathering? No, no, no. The complete, like I just showed you, complete analysis can be done from the R package. And it has the nice feature, and I'm just showing to you this here. So here I'm in the R session, right? You have this open Reactome. So if you do the analysis from the R session, you actually have this feature, say open Reactome, and it opens the pathway browser. So you can jump from your R session to the pathway visualization of the pathway browser, and from the pathway browser to your R session. So both ways are possible. Does Reactome work fine too for single-cell RNAseq? It's just what I showed you. Is Reactome runnable in R? So runnable, yes, we have this R package, but it uses the web service. For phosphoproteomics data, the identifier first column will be protein implies phosphosite. No, unfortunately, we don't support phosphosite because there's no standard for that, first of all. So no standard way to annotate phosphosites, and we don't have the phosphosite level data. We have it, but we can't incorporate at the moment in the pathway analysis. Can we use already calculated log fold changes and p-values to see Reactome visualizations? Unfortunately not. No, we need your original data. And the last question I'm going to answer now, since long read platforms produce fewer counts per gene than short read platform, does that reflect in the pathway analysis? No, that's the very neat way. Since so if you have different data sets, so that of course the same technology has to be used within one data set, but then it does not affect your data since you stay within the system, right? So even though you have more coverage, you still compare to the same thing. So, and then you normalize it on pathway level. So no, this is not a problem. So now I'm heading back to my slides. So I want to thank everyone who works on ReactomeGSA or worked on them, all of my colleagues at the EBI and at the Medical University in Vienna. And additional resources. So you can find the pathway browser at www.reactome.org. You can find all the examples that I showed you very briefly as Jupyter notebooks on reactome.github.io/ReactomeGSA. So there we have a whole collection of tutorials that you can have a look at to see a detailed usage of the R package. The R package is ReactomeGSA on Bioconductor. There, of course, we have vignettes to how to analyze single-cell data and do multi-omics pathway analysis. So you have all the resources there. But again, please don't hesitate to contact us on help@reactome.org.